Hello everyone. In this lecture video, we're going to talk about recombinant DNA. First, let's talk about the objectives. In this video, you should learn about restriction endonucleases, what they are, and why they are important for studying the structure of DNA. You should learn about the steps to purifying plasmid DNA from a culture of bacteria. You should also understand how restriction enzymes are used to cleave plasmid DNA and how those fragments can be separated using gel electrophoresis. You should also be able to read gel electrophoresis to determine the length of DNA fragments. So first, let's talk about what a plasmid is. A plasmid is going to be a type of DNA that is oftentimes found in most bacteria. It is a small, circular, double-stranded DNA molecule. This is separate from the bacterial chromosomal DNA. As you can see here, all bacteria are going to have this chromosomal DNA. However, they don't all have this plasmid DNA here. So this is sort of like an accessory type of DNA that bacteria have. It is separate from the chromosomal DNA. However, as bacteria replicate, as they divide and produce more of themselves, this plasmid DNA will also get replicated. And like chromosomal DNA, plasmid DNA also has the ability to express itself. This is oftentimes used in biotechnology because plasmids can be engineered to study particular genes of interest, and it is also a good way to amplify a gene of interest. So if you insert a plasmid that has been genetically engineered with the gene that you want to study, you can easily amplify this gene by letting the bacteria grow. Usually this plasmid is going to be engineered with a particular type of antibiotic resistance. So for example, ampicillin resistance can be used in this plasmid. And this is important because it will separate the bacteria that have this plasmid from the ones that don't have it. For example, if you have bacteria that have this plasmid and some that don't, what you're going to do is place this plasmid on a petri dish with nutrients and also with the antibiotic. The bacteria that have this plasmid and therefore the antibiotic resistance gene will multiply and grow on this petri dish, and the ones that don't have it will die off. So this is a good way to separate the bacteria that have the plasmid from the ones that don't. Plasmids often are used to carry small pieces of foreign DNA. So what will happen is here is a gene of interest, and it is going to be inserted into this plasmid. However, what you need to keep in mind is that the insertion happens by base pairing over here. I should also mention that this base pairing to happen, for this pasting to happen, an enzyme called ligase is needed. So when you have this plasmid that has the antibiotic resistance gene and has the particular gene that you want to study, this is going to be called a recombinant DNA. DNA is oftentimes cut with a restriction endonuclease. This is also called a restriction enzyme. This is an enzyme that is naturally found in bacteria. Restriction enzymes help defend bacteria against viral infections. As you can see in this diagram here, you have a bacteria, and here is a phage, and a phage is a bacterial virus that is infecting the bacteria, and it is injecting its genetic material into the bacterium. What this bacteria will do is to release these restriction enzymes that would cut up the pieces of the DNA that are from the virus so that it cannot be infected with this virus. And interestingly enough, this whole process of methylation will protect the chromosomal DNA of the bacteria. Now, restriction enzymes don't just cut anywhere, they cut at specific nucleotide sequences called restriction sites. So the restriction site really depends on the type of restriction enzyme involved. Different restriction enzymes will cut at different restriction sites. And some pieces of DNA will have multiple restriction sites, and some will have only one. So a recombinant DNA will include genes of interest that we want to study, and also an antibiotic resistance gene that will help these particular types of bacteria survive and replicate. And this cutting right here of the DNA in both the plasmid and our DNA of interest happens by this restriction enzyme that will cut at a specific sequence of DNA. And when these pieces of DNA are added together and ligase is added, you can have what is called a recombinant plasmid. Again, these pieces will come together by complementary base pairing. Let's look at this diagram over here. Here you could see a plasmid DNA on top that has already been genetically engineered with ampicillin resistance. And here we have an isolated gene of interest. You could see that the cut DNA of both of gene of interest and plasmid DNA with the same restriction enzyme. These two pieces of DNA will come together with base pairing and also with the use of ligase. And what you're going to end up here is a recombinant DNA plasmid. Next, you're going to have these bacteria and the plasmid placed together in a test tube. And what's going to happen is that heat shock is going to be applied to this test tube. Now, some of the bacterium will take up the plasmid and some won't. And that is why at this step, at this point, it is important to have this antibiotic resistance gene. It will help get rid of the bacteria that have not picked up the plasmid. The bacteria are going to then be placed in this petri dish that is going to have nutrients that will help the bacteria grow, but it will also have antibiotics. And in this case, it would be ampicillin. So the bacteria that end up growing in this petri dish are going to be the ones that have the plasmid DNA, which has the ampicillin resistance gene, and the ones that don't have this plasmid don't have the ability to resist the antibiotics, and they will die off. The bacteria can then be collected and studied, and the plasmid can be purified for further studies. So I'm going to talk about the procedures that you have to follow to purify plasmid DNA. But first, let's talk about what a centrifuge is. In molecular biology, in microbiology, and in chemistry, you are often going to use this device here called a centrifuge that is going to spin samples really, really fast and separate out different components that are within a liquid. What you need to keep in mind when you use the centrifuge is that it always needs to be balanced. So what that means is if you put 2 milliliters of bacteria here, you want to make sure you put another test tube right across from it that is going to be 2 milliliters. And even if you don't have a sample, like another sample that you want to centrifuge, it does not matter. You always want to make sure that your centrifuge is balanced. So let's say you have 2 milliliters of bacteria, you want to make sure you put 2 milliliters of water, for example, on the other side of it. But anytime you use the centrifuge, it has to be balanced. Otherwise, it's going to start to shake and fall off your table. So these are the steps of isolating plasmid DNA. You can't do them because you're not in lab at the moment, but I'm going to do my best to explain every step and why you have to go through each step in order to isolate the plasmid DNA. So first, you're going to take 1.5 milliliters of bacterial culture right here, and you're going to centrifuge it for 30 seconds. And when you centrifuge it, what you're going to get is this pellet at the bottom over here. This pellet, this is what you want to keep. These are the bacterial cells. And then in this step over here, this supernatant right here, the liquid portion, is something that you don't need anymore. This is the growth media and the antibiotics. So this is the first step of separating the cells from the other stuff that was in that petri dish or in that bacterial culture that you no longer want. And according to your lab manual, you should do this twice. So you should add another 1.5 milliliters of bacterial culture to the same tube and go through the same step of centrifuging and allowing the bacteria to separate from the other components. You will need to get rid of the supernatant here, and you could use a pipette for that. Next, you're going to add 600 microliters of TE buffer or water to the cell pellet and resuspend completely. And all the pellet that you had at the bottom of the tube should be resuspended. And this is because you don't want all of your bacterial cells to be sitting in one corner of the tube. You want them to be redistributed in this liquid. And you're going to shake it, and everything should be resuspended. Next, you're going to add 100 microliters of cell lysis buffer. Remember, we're trying to isolate the plasmid DNA, so we are going to have to lyse the cell. So when you add these 100 microliters of cell lysis buffer, you're going to mix by inverting the tube 6 times. So you're just going to turn it 6 times, but do not shake your tube at this point because you could damage the DNA. And also, you shouldn't let this sit for too long because it is going to, again, damage parts of the DNA that you want to keep. You're going to see that the solution color is going to change from opaque to clear blue, indicating complete lysis of the cell. This lysis buffer contains sodium hydroxide and detergent, STS, which is going to lyse the bacteria. And also the pH is going to be very high, which is going to denature the chromosomal DNA and the plasmid DNA, which is okay because the plasmid DNA can be renatured. So here we are, we're kind of at this step over here where we have the cell extract in our test tube. You're going to add 350 microliters of cold neutralization solution. And this neutralization solution is going to return the pH back to neutral. And you're going to mix by inverting the sample 3 times to ensure complete neutralization. And your test tube, the color of the liquid in your test tube should no longer be blue. And you're going to centrifuge this again at maximum speed in the microcentrifuge for 3 minutes. What's going to happen is that the DNA of the plasmid is going to get renatured, but that long chromosomal DNA is going to get tangled with other cellular components that you don't want anymore, and they're going to form a insoluble precipitate. So at this point, what we want is the supernatant, because that's where the plasmid DNA is going to be, and whatever is at the bottom of the tube is the other components of the cell that we want to get rid of. So you're going to transfer the supernatant to a pure yield mini column, which is this over here. And this is the mini column. This is over here. And do not disturb the cell debris pellet. For maximum yield, transfer the supernatant with a pipette. You're going to place the mini column into a pure yield collecting tube and centrifuge at maximum speed in a microcentrifuge for 15 seconds. So here's the mini column, and here's the pure yield collecting tube. So what's going to happen here is that the mini column contains a silica membrane right here in this portion that will tightly bind the plasmid DNA. So the plasmid DNA is not going to get past it. It's going to remain in this mini column over here. But the other macromolecules that are remaining are going to be centrifuged through the tube over here. So the plasmid DNA will stay here, and the other things that you don't need are going to be found down here in the tube after you centrifuge them. Next, you're going to add 400 microliters of column wash solution to the mini column and centrifuge again at maximum speed in a microcentrifuge for 30 seconds. It is not necessary to empty the pure yield collection tube. And the whole point of this is to remove any leftover salts from the buffer. Then you're going to transfer the mini column to a clean 1.5 microcentrifuge tube, and then add 30 microliters of elution buffer to the mini column matrix and let it stand for 1 minute at room temperature, and this will allow the DNA to become solubilized. And then we're kind of at this point over here where you're going to have purified plasmid DNA. And you're going to centrifuge at maximum speed in microcentrifuge for 15 seconds. And then you're going to have the clear, colorless liquid at the bottom of the centrifuge tube that contains purified plasmid DNA. So this is a video that I found that goes through all the steps. It shows you how it's done in lab, step by step. It is identical to what is in your lab manual. The measurements are slightly different, but the steps are exactly the same. You will see each step done in this video and each step explained. And I highly recommend that you also watch this video because the steps and understanding how to purify plasmid DNA can appear in your lab practical and your quizzes. So once we have this purified plasmid DNA, we want to use different restriction enzymes to see what type of DNA is in this plasmid. So the steps are pretty simple, and I have another YouTube video here that goes through all the steps. So you're basically just going to pipette 5 microliters of DNA into an empty 1.5 milliliter centrifuge tube. You're going to add the appropriate restriction digest buffer. You're going to write down the name of the restriction enzyme that you're going to be using. And you're going to add 2 microliters of your assigned restriction digest. Again, it could be any, it depends on what you're assigned. And you're going to bring up the volume to 20 microliters by adding 11 microliters of water to the reaction and letting this tube incubate at 37 degrees. This could be in the water bath, or it could be in an incubator. So what will happen here is that the restriction enzyme will cut the DNA on specific restriction sites. There could be one, there could be multiple, it all depends. What you're going to end up with is DNA fragments. And DNA fragments can be separated by this instrument, this lab instrument here, called gel electrophoresis. And this is very commonly used and very important in molecular biology. What you have here is this container here with gel inside of it. This is agarose gel. The gel is heated up and it's poured into this container and it's cooled. And the unique thing about this gel is that it is porous. It has tiny little holes in it. And macromolecules can travel in these tiny little holes. And depending on how far they travel, they tell us something about the size of the macromolecule, but also about how compact the macromolecules are. So DNA fragments can be separated according to their length by gel electrophoresis. If you have a long piece of DNA, it is not going to travel very far. if you have a short piece of DNA, it's going to travel pretty far. DNA carries a net negative charge, and it will migrate towards the positive pole of the electric field. So you're going to place the DNA in these wells called towards the cathode side, towards the negative side. And a current is going to be applied to this gel. So how far these DNA molecules travel after the current is applied to them will tell you the relative size of these DNA fragments. And this separation allows researchers to determine the correct DNA fragments to insert into plasmid, but also it could be used for verification purposes. What you're going to have in this gel electrophoresis is a negative side and a positive side. The negative side is called the cathode, and the positive side is called the anode. And for DNA, you need to load it on the cathode, on the negative side, because then DNA will want to move towards the positive side when the electricity is applied. Now, when I was learning about this, I was always confused because I was like, why is the negative side called a cathode? And why is the positive side called an anode? What you need to think about is this way. Anions want to move towards the anode, and cations will move towards the cathode. Okay, so let's kind of look at how the gel is going to be read. You're going to have these wells at the top over here where you're going to load the DNA on. And then they're going to, and an electric field is going to be applied, and they're going to travel depending on their size. Now, this far left column here, it's just usually like a reference point. It kind of tells you like how many kilobase pairs, for example, a piece of DNA is if it came this far. So you could think about this sort of like as a ruler or, you know, a reference point for measurement. These are called columns, and by the way, these over here, these lines that you see that form are called bands. So again, DNA is a negative molecule, and it is going to move towards the positive end of the gel when the current is applied. And I want to mention this here, if it's a large molecule, it won't travel very far, and if it's small, it will travel further down the gel. So let's look at this example over here. Okay. So here's a plasmid DNA over here with these restriction sites. Below is a restriction map of plasmid PGen 101, which is 20 kilobase pairs. Using the map as a guide, give the number of restriction fragments along with their associated length that would result from digesting PGen 101 with restriction enzyme EcoR1, BAMH1, and a combination of EcoR1 and BAMH1. So first, let's look at this enzyme, EcoR1. So if we mix this plasmid with EcoR1, we could see that EcoR1 has only one restriction site on this plasmid, and that is right here. So it will cut this plasmid DNA right here and nowhere else. What's going to result is a linear piece of DNA, but it is going to be the same length. It's still going to be 20 kilobase pairs. So if you load it onto the gel here and apply an electric field to it, what you're going to see here is a band forming right here at 20 kilobase pairs. That's what's going to happen. Now, what if we added this plasmid with BAMH1? If you look at this plasmid, BAMH1 on this plasmid has 3 different restriction sites. One over here, and one right there. And we're going to ignore EcoR1 because we're just talking about BAMH1. You're going to end up with 3 fragments of DNA. One right here, from here to here, which is going to be 12 kilobase pairs, one that is going to be 6 kilobase pairs, and then one that is going to be 2 kilobase pairs. If you load this, and I should make this blue so it's not confusing. If you load this, pour this into the well over here, and you apply electricity to it, you're going to find one that's going to be about 12 kilobase pairs, so right here. 2 kilobase pairs, oops, right here, and one that is going to be 6 kilobase pairs, which would be like right around here. So that's what you're going to see, 12, 6, and 2. Now, what if we added EcoR1 and BAMH1 together to this plasmid DNA? So, so all the restriction sites that we see here, you're going to have one cut over here, one over here, one over here, and one over here. So you're going to end up with 4 fragments of DNA, one that is going to be 8 kilobase pairs, and will form a line right here. One that is going to be 6 kilobase pairs over here. One that's going to be 4 kilobase pairs, about right here. And then 2 kilobase pairs over here. So this kind of should give you an idea of how restriction enzymes work with restriction sites and how the length of DNA fragments can be calculated using the gel. Now, as you saw in all the procedures that I just talked about, you're going to need very precise measurements of different solutions. And you will need to learn how to use a micropipette. So I will do my best to explain it in a video. This is something that is going to take some practice. So when you use a micropipette, you should never use it by itself. You have to always use a disposable plastic pipette tip. So it will look something like this over here. So don't ever dip this tip area into any kind of liquid. The way that the liquid will get soaked up, or the pipette will pick it up, is that first you have to push down this button over here, and you're going to feel resistance. There's going to be two stops with this button, but whenever you want to pick up the liquid, you want to stop at the first resistance that you feel. You're going to stop at the first resistance. You're going to insert the micropipette into your liquid and gently release the button. And the liquid will be picked up by the pipette. Now, when you want to eject this liquid, what you're going to do is press down on this button again, but you're going to go past the first stop. You're going to go past the first resistance, and you're going to push down to the second resistance. And that's how the liquid is going to be released from the pipette. And this illustration here just kind of shows you have to make sure you have the pipette tip. You're going to push down to the first resistance. You're going to insert the tip into your liquid, let go of the button, and then when you're ready when you want to eject the liquid, you're going to push down this button past the first stop and go all the way down to the second stop, and the fluid will be released. And you always want to make sure that you hold the pipette vertically. Some precautions to keep in mind with the micropipette is that you should never ever try to set a pipette above or beyond the volume that it is set for. So each pipette that you work with is made for a certain volume. You should never go above or below that. If you do, you could damage the pipette. You should always apply the pipette tip before using the pipette. And the pipette should always be held down vertically. you should get some practice with controlling the release button. You shouldn't do it too fast. And also, when collecting liquid, hold down the pedestal to the first stop. So this is when you want to pick up liquid. And then you're going to immerse the tip in the liquid. And always change the pipette tips before uses. So let's look at different micropipettes. The name tells you something about the maximum volume, pretty much, that it can hold. So P10 can hold maximum 10 microliters. P100 can hold a maximum of 100 microliters. And P1000 can hold a maximum of 1000 microliters. You should keep these ranges in mind in lab and know which pipette is appropriate for the volume that you need. You need to understand how to set the volume on the micropipettes. So with P10 and P20, they're kind of similar. What you need to think about, you have 3 digits over here. There is a decimal right here. So this is 2.5. So if this can hold a maximum of, P10 can hold a maximum of 10 microliters. So this would be 10, and there's a decimal here, and this is 0. So if you have 2.5, this would be 2.5, and there's no digit here. And the same goes with the P20. So this would be, if you had 20 microliters, this would be 200, because there would be a decimal here. If you have 18.2, it would be 18.2. Now with P200, it will be kind of different. So for example, here 110 would just be 110. The decimal, there is no decimal place here. With P1000, it's kind of tricky. You have to think about it this way. You're just going to skip the last digit. So for 1000 microliters here, it would just be 100. And we're pretty much leaving out the last digit. For 710 microliters, it would be 071, and then the last 0, we're just going to leave it out. So this is something that will take a little bit of practice to understand. I do have this really good video here that will explain micropipetting to you in more detail. So please be sure to also watch this video. It will teach you more about how to hold it, how to use it, and how to set the digits properly.